Hello friends, today we will talk about analog versus rational approach of drug design. The main objectives of today's deliberations are to give a brief introduction about drug design, to get knowledge about drug analogy, to study in detail about rational drug design approach, and to know about the factors governing drug design. So, first of all, drug design. Drug design, often referred to as rational drug design or simply rational design, is the inventive process of finding new medications based on the knowledge of a biological target. The drug is most commonly an organic small molecule that activates or inhibits the function of a biomolecule, such as a protein, which in turn results in the therapeutic benefit to the patient. In the most basic sense, the drug design involves the design of molecules that are complementary in shape and charge to the biomolecule target with which they interact and therefore will bind to it. Drug design frequently but not necessarily relies on computer modeling techniques. This type of modeling is sometimes referred to as computer-aided drug design. Finally, drug design that relies on the knowledge of the three-dimensional structure of the biomolecule target is known as structure-based drug design. In addition to small molecules, biopharmaceuticals and especially the therapeutic antibodies are an increasingly important class of drugs, and computational methods for improving the affinity, selectivity and stability of these protein-based therapeutics have also been developed. The phrase drug design is to some extent a misnomer. A more accurate term is ligand design, that's the design of a molecule that will bind tightly to its target. Although design techniques for prediction of binding affinity are reasonably successful, there are many other properties such as bioavailability, metabolic half-life and side effects that first must be optimized before a ligand can become a safe and efficacious drug. These characteristics are often difficult to predict with rational design techniques. Now, the analog approach of drug design. The term analogy, derived from the Latin and Greek analogy, is used in natural science since 1791 to describe structural and functional similarity. Extended to drugs, this definition implies that the analog of an existing drug molecule shares chemical and therapeutic similarities with the original compound. The chemical design of analogues makes use of simple and traditional procedures of medicinal chemistry, such as the synthesis of homologues, analogues, isosteres, positional isomers, optical isomers, transformation of ringed systems, and synthesis of twin drugs. Analogues produced by homologous variations, which includes homology through monoalkylation, simple homologous variations applied on a neuroamidase inhibiting lead achieved a 6,300-fold increase in potency. Polymethylenic base ammonium compounds, that includes hexa and decamethonium compounds, usually show high affinity for the cholinergic receptors. When the values of N are intermediate, that's N is equal to 5 or 6, penta or hexamethonium, these compounds behave like cholinergic agonists towards the sympathetic ganglion. For higher values, that's N is equal to 10, that is decamethonium, the compounds become antagonists of acetylcholine at the muscular end plate. In both cases, increasing acetylcholine levels displace them from their binding sites. Homology in cyclic compounds. Homology in cyclic compounds can dramatically change the affinity of a ligand for its target. Analogues produced by analogy Tolcapone was designed as an inhibitor of the enzyme catechol O-methyl transferase and is useful in the L-DOPA treatment of Parkinson's disease. In avoiding the methylation of L-DOPA as well as that of dopamine, it prolongs the beneficial activities of these molecules. Catechol O-methyl transferase inhibition represents therefore a valuable adjuvant to the L-DOPA decarboxylase inhibition. Unfortunately, tolcapone exhibited severe liver damage and had to be removed from the market. The corresponding analogue entacapone is devoid of this side effect. Analogues produced by isosteric variations. The dominant parameter is structural. Structural factors are important when the portion of the molecule involved in the isosteric chain serves to maintain other functions in N. For the two antidepressants, the bioisosterism is geometrical insofar that the dihedral angle formed by the two benzo rings is comparable. A is equal to 65 degree for the dibenzopyene and A is equal to 55 degree for the dibenzocycloheptadine. This angle is only 25 degree for the neuroleptic phenothiazines and for the thioxanthines. The dominant parameter is electronic. The electronic attracting nitro group in the antibiotic chloramphenicol has been replaced by other electron attracting functions such as methyl sulfonyl or an acetyl. Apparently, the dominant feature is of electronic nature. Similar electronic effects are found in the benzodiazepine series, in which the chlorine atom of diazepam can be exchanged with bromine or with a nitro group, a particular geometry. The dominant parameter is lipophilicity. Typical examples of lipophilic analogues or prototypes are epistamine, derived from physostigmine by replacement of the N-methyl group by a N-heptyl group, and tiagabine, in which a diaryl butynyl chain is grafted to the nitrogen atom of nicotic acid in order to yield a compound able to cross the blood-brain barrier. Analogues produced by ring transformations. When active molecules contain cyclic systems, these can be opened, expanded, contracted and modified in many other ways or even abolished. Conversely, non-cyclic molecules can be cyclized, attached to or included in ring system. Two interesting examples are found in cyclic analogues of B blockers. Cyclization to the morpholine yields the antidepressant vilozane, whereas cyclization to chromanols yields the potassium channel blocker, that is chromocalin. Twin drugs. The search for cationic cholinergic agents has led to numerous twin drugs. The bis-quaternary ammonium salts, that is hexamethonium and decamethonium, are potent blockers in ganglia and in neuromuscular junctions respectively. Other neuromuscular blocking agents such as succinyl and sebacetyl dicholine can be regarded as pure acetylcholine twin drugs. For example, hexamethonium and decamethonium. Now, the pros and cons of analog design. The usual objectives of analog design are the identification and development of a possibly improved version of a prototype drug. Such compounds are often direct analogues and therefore are chemically and pharmacologically similar to the prototype drug. It should also present some advantage over the prototype. However, to achieve these goals is not always an easy task. Analog design as a means of discovering new drugs. New use for old drugs. In some cases, a new clinical activity observed for an old drug is sufficiently potent and interesting to justify the immediate use of the drug in the new indication. And this is illustrated below. Amiodarone was introduced as a coronary dilator for angina, but concern about corneal deposits, discoloration of skin exposed to sunlight, and thyroid disorders led to the withdrawal of the drug in 1967. However, in 1974, amiodarone was found to be highly effective in the treatment of a race type of erythema known as the Wolff-Parkinson-White syndrome. Accordingly, amiodarone was reintroduced specifically for that purpose. Benzodiazepine was initially used in Europe as a coronary dilator and proved later to be useful urosoric agent. However, it is now withdrawn from the market due to several cases of jaundice associated with its use. The corresponding brominated analogue, benzobromarone, was specifically marketed for its urosoric properties. Newlets from old drugs. The SOSA approach. The SOSA approach, that is selective optimization of site activities, represents an original alternative to high throughput screening, that is HDS. SOSA consists of two steps. Perform screening assays on newly identified pharmacological targets only with a limited set, that's approximately 1,000 compounds of well-known drug molecules. For these drugs, bioavailability and toxicity studies have already been performed and they have proven usefulness in human therapy. And second is, once a hit is observed with a given drug molecule, the task is then to prepare analogues of this molecule in order to transform the observed site activity into the main effect and to strongly reduce or abolish the initial pharmacological activity. Now the drug design, a rational approach. Drug design or tailor-made compound aims at developing a drug with high degree of chemotherapeutic index and specific action. It's a logical effort to design a drug on as much as rational as possible, thus reducing to the minimum the trial and error approach. It essentially involves the study of biodynamics of a drug, besides the interaction between drug molecules and molecules composing the biological objects. Rational approach to drug design. A rational approach to drug design may be viewed from different angles, namely quantum mechanical approach. Quantum mechanics or wave mechanics is composed of certain vital principles derived from fundamental assumptions describing the natural phenomena effectively. The properties of protons, neutrons, and electrons are adequately explained under quantum mechanics. The electronic features of the molecules responsible for chemical alterations form the basis of drug molecule phenomenon. Second is molecular orbital approach. Based on the assumption that electrons present in molecules seem to be directly linked with orbitals engulfing the entire molecule, which set forth the molecular orbital theory. The molecular orbital approach shows dependence on electronic charge as evidenced by the study of three volatile inhalation anesthetics, and also on molecular conformation as studied with respect to acetylcholine by such parameters as bond lengths and angles including torsional angles. Molecular orbital calculations are achievable by sophisticated computers, and after meticulous interpretations of results, the molecular structure in respect of structure activity analysis is established. Third one is molecular connectivity approach. This approach establishes the presence of structural features like cyclization, unsaturation, skeletal branching, and the position and presence of heteroatom in molecules with the aid of a series of numerical indices. For example, an index was determined to possess a correlative factor in the SCR study of amphetamine type hallucinogenic drugs. Molecular connectivity approach has some definite limitations such as electronegativity variance between atoms, non-distinguishable entity of cis-trans isomerism. And fourth one is linear free energy approach. This method establishes the vital link between the proper selection of physicochemical parameters with a specific biological phenomenon. However, such a correlation may not guarantee and allow a direct interpretation with regard to molecular structure, but may positively offer a possible clue towards the selection of candidate molecules for synthesis. Now, the drug design, the method of variation. Under this method, a new drug molecule is developed from a biologically active prototype. The various advantages are as follows. At least one new compound of known activity is found. The new structural analogues, even if not superior, may be more economical. Identical chemical procedure is adopted and hence considerable economy of time, library and laboratory facilities. And fourth, screening of a series of congener, that is member of the same gene, gives basic information with regard to pharmacological activity. Fifth one, similar pharmacological technique for specific screening may be used effectively. The cardinal objectives of the method of variation are to improve potency, to modify specificity of action. The applications of the method of variation, depending on the considerations enumerated above, is exploited in two different manners to evolve a better drug. The two main approaches for this goal can be indicated as drug design through disjunction and drug design through conjunction. Drug design through disjunction comes in where there is the systematic formulation of analogues of a prototype agent, in general towards structurally simpler products, which may be viewed as partial or quasi-replicas of the prototype agent. The method of disjunction is usually employed in three different manners, namely unjoining of certain bonds, substitution of aromatic cyclic system for saturated bonds, diminution of the size of the hydrocarbon portion of the parent molecule. Now, the drug design through conjunction. This is known as the systematic formulation of analogues of a prototype agent, in general towards structurally more complex products, which may be viewed as structures embodying in a general or specific way certain or all of the features of the prototype. In this type of drug design, the main principle involved is the principle of mixed moieties. A drug molecule is essentially made up with two or more pharmacophoric moieties embedded into a single molecule. Now, the tailoring of drugs. With the advent of enormous in-depth knowledge of modern chemistry, the tailoring of drugs has become a skillful art that may result fruitful results through specific modes of attack on a drug molecule. Various conformational and stereochemical chains afford flexibility and overall dimension of a drug molecule. Such alterations may be conveniently achieved through different means and ways, namely ring fission or fusion, formation of lower or higher homologues, introduction of optically active centers, formation of double bonds towards geometrical isomerism, and lastly, introduction of bulky groups towards restricted rotation or the removal and replacement of such groups. Alterations of various physical and chemical characteristics through the insertion of newer functional moieties are by the replacement of such groups already present by others that essentially differ in degree or in type. These types of change may be effectively brought about by isosteric replacement, change of orientation or position of given moieties, introduction of polar character of given functional groups or replacement of other groups with different electrical features. And finally, such changes which either promote or inhibit the presence of different electronic conditions achieved through inductive effects, mesomeric effects, tautomerism, chelation, hyperconjugation. So this brings us to the end of the lecture. Hope you have enjoyed and understood. Thank you and goodbye.